All right, so now it's my pleasure to introduce our first team talk of the day. This is a team that's going to present new findings from a project to fully reconstruct the morphology of genetically targeted neurons and their detailed axonal projections throughout the brain. The talk is titled The 3D Reconstruction of Single Neurons at the Whole Brain Level. And this will be presented by Yoon Wang, Stacy Sorensen, Ji Zhao, Peng Si, and Yang Li. Please join me in welcoming them.
So here we are reporting the 3D single neuron reconstruction at the whole brain level. This is a full morphology project that we have been performed for nearly 3 years. Why full morphology reconstruction? By doing the 3D whole brain level single cell reconstruction, we believe that we can answer some crucial questions that has been raised in the neuroscience field over a century. Such as what's the specificity and the diversity of projection targets at the single cell level, and how does local morphology relate to long-distance projection patterns, and how does full morphology specificity relate to cell type classification. The methods we use mainly include 4 parts. Different lines of transgenic mice with sparsely labeled neurons, the brain fixed, then sent to host in China using FMOST technique to get the image data. Briefly, the FMOST technique is like using two-photon microscope to scan the high-resolution image with very thin, about 1 micrometer thick. Then cut the layer off, then continue to scan. In this way, they can get the whole brain image stack at the, to finish the whole brain, which the data size about 10 TBs. Then we use the big data to do the single cell reconstruction at the whole brain level. Initially, we use Neolucida 360. Recently, we changed to a more advanced 3D VR. Also, we have auto-tracing algorithms in progress. And subsequently, various bioinformatics analysis will be performed. As you may know that the 3D single-cell reconstruction at whole brain level was very difficult like 10 years ago, and the study very rare because the method is complicated. The neurons has to be filled in vivo, then the whole brain cut it in serial sections. Then the single neuron tracing has to be traced from like more than 100 serial sections. One neuron tracing may took about several months to finish. So that was impossible to carry out any systematic study on any specific type of neuron. In recent years, more and more study has been reported on the whole brain level single-cell neuron reconstruction. However, if you take a look closely, you will notice that most of the neurons are incomplete by missing local branches of the axons largely. Because of the initial branching was missing, then some long projections also missing. We are able to get complete neuron taking advantages of 2 major tools. One is the transgenic line, different line of transgenic mice, which allowed us to do carried out a systematic study on a specific type of neuron. And recently we have the VR added to VAS3D, and then which allowed us to directly visualize and tracing the complex and fine structure in a virtual space, which is much more accurate than any other tracing tools so far available. here, just showing you some examples. This is only 13 claustrum neurons. Already you can see it largely outlined two cortical hemispheres. The claustrum is a structure which is narrow and thin, and the white matter, which have connections almost to all the functional cortical regions, and also connect to some subcortical regions. Here is 5 claustrum neurons, just showing you the zoom-in fine structures. And this is 8 and 3 layer 6 BPCs. I would say we are kind of approaching the ultimate level for single neural morphology. By using the GMB4 transgenic mass line, we carried out our first full morph project. In this transgenic line, the two major populations were labeled. One is the claustrum neuron, the spiny claustrum neuron, and the other is the layer 6 BPCs. We found 4 morphology type, actually 2 major type and 2 subtype each, according to their projection mainly on the midline as a one major type, and avoid the midline as the second major type. According to the project only in one hemisphere or bilateral 2 hemisphere classified subtype A and B. So it's very interesting that we found, although the claustrum very small structure, but still we can see the axon morphology varies depending on the soma location at anterior or middle or posterior part of the claustrum. So this is present a topographic organization. The layer 6B also can be classified into ipsilateral and bilateral according to their axon morphology projection in one hemisphere or 2 hemisphere. You can also see that the axon morphology also varies depending on their soma locations. So these 2 very different populations, they both present the tomographic organization features. Stacy will introduce our second full morph project.
Hi, I'm Stacy Sorensen. I run one of the morphology groups here at the Institute. We are primarily focused on reconstructing cortical neurons labeled in a slice, and so to be able to look at these full intact morphologies is a real gift, and I think will change how we look at the brain. So today I'm going to be talking about LGN neurons, and as many of you know, this brain structure is one of the more well-studied structures in the brain across many species, but there are still a number of open questions, particularly in LGN in the mouse, such as, are there distinct types of relay neurons in LGN? Are there morphological correlates of LGN subregions, such as the shell and the core? And then do thalamocortical projections correlate with described functional circuitry? And then a little more specifically, now that we have these full intact neurons that were labeled in vivo, we can ask, revisit questions like, are there distinct thalamocortical projections from the shell and the core? And then are there multiple dendritic types, as has been described in the slice? And then do we see morphologies and projection patterns that relate to these previously described functional domains in LGN? And then this is a video, yes, that's going to play. Fantastic. So I just want to show you the kind of labeling that we're working with in order to generate these 3D reconstructions. So you want to kind of follow this area here. You see those, there they were, and they're gone. So there's about, basically, but just to show that there's about 120 neurons labeled in these TNT1A brains that are crossed to a couple of different transgenic reporter lines, and we can very faithfully follow get the local morphology and also the long-range projections of these LGN neurons. Here I'm just showing all the LGN neurons that we reconstructed from two different TNT1A brains and then mapped to the CCF, or the Common Coordinate Framework, and then Yang will be talking in a minute about how this mapping is done. Then I have further divided the LGN neurons into these different domains of LGN, the core, this should say the ipsilateral zone, and then the shell. And you can kind of get a sense from just looking at this overview that these core neurons have these quite bushy axon terminals in cortex and span much of the cortical layers. A similar structure comes with the ipsilateral neurons, and then shell neurons are just looking a little different, though of course at this point the data set is fairly small. Just to give you a sense about the different types of neurons that we're seeing based on the morphological and projection target diversity. So for the LGN-core, we are mostly seeing neurons that have this Y-type morphology, where you have dendrites that project radially around the soma. And then I'm showing a coronal and then a horizontal view of the neurons. And then if we look at their projection targets, they're mainly in primary visual cortex, or VIS-P, but we do have a couple of neurons that have this Y-type morphology that also send axons to the higher visual areas, in this case VIS-L, and then we have one example of a X-type neuron based on the dendritic morphology that's much larger in comparison to the Y-type cells, and that primarily goes to these higher order visual areas. If we look at our small population of shell neurons, we can see that they all have this W-type morphology, where the dendrites are oriented to one side of the soma. And then if we look at their projection targets, they're mostly contacting higher visual areas and retrosplenial cortex. So using this very preliminary data set of brain-wide morphologies, we observe an increased diversity beyond what's been previously described, but of course our N's are still very small. And this is just one final slide that I want to visit this question of, at the single cell level, are there distinct thalamocortical projections that correlate with these functionally distinct pathways that have been previously described? A direction selective pathway that runs from the retina through the shell, that goes selectively up to these superficial layers, versus this non-direction selective, more classical pathway that should go through the shell, and then with axons ending in layers 4 and deeper layers. So here I just have one example of a shell neuron and one example of a core neuron with the axon colored by layers according to the CCF. But so we know that this is something that we want to do some further validation on, but just to give you a sense that most of the LGN neurons that we're seeing have axon that's going to layer 1. It's seeming like the relative distribution of layer 1 axon for core neurons and shell neurons may be different, and then also the axon itself looks quite different. So we're looking forward to collecting many more of these full neuron reconstructions, and then this work that I'm describing here has been mostly qualitative, but we know we want to move to a more objective quantitative analysis, and that's what Peng will be presenting on. So, thank you.
Hello, everyone. My name is Peng. I joined the morphology team this summer, and my role here is to perform quantitative data analysis. Specifically, I've been working on objective cell typing using the whole brain full morphology data. And in today's talk, I will use this genetically sparsely labeled mouse brain 17302 as an example to demonstrate our methodologies. This is the first mouse brain that we fully reconstructed, and it includes over 100 neurons. So through analysis, we want to answer the following questions. How many morphologically distinct cell types can we identify? How are these morphological features related to projection patterns? And finally, what's the relationship between morphology and functionality? So here's an overview of this data set. As you can see, it includes thalamic neurons, especially the LGN neurons, and the striatum neurons, which is also called CPU neurons. These data are generated according to a protocol that includes several levels of readiness. At level 0, we locate the soma position, dendritic area, and the axon terminal projections. At level 1, we fully reconstruct the dendrite and also trace the major branches of axons. At level 2, we complete the reconstruction at axon distal clusters. And then computational analysis kick in by starting with post-processing steps to ensure that every neuron is represented by a fully connected single tree. And all the computationally generated connections will be labeled as well as the unconnected branches for further check. So neurons with more than 10% unconnected branches will be excluded from further analysis. Here's an example showing our level 2 reconstruction. And in the future, we'll also proceed to a level 3, which will recover from the raw image, the spine, boutang information, and more. So these reconstruction data are then passed to the next step for quantification of features. With full morphology, we can now expand our study to the long projection axon shafts as well as the distal axons. Actually, features from these regions turns out to be very useful for classifying different types of cell types. As you can see that these long axon features and distal axon features showed high mutual information. We also performed feature-wise comparisons and observed significant correlation or anti-correlation between these features, which also indicate certain level of redundancy. So to reduce this redundancy, we performed principal component analysis to reduce the number of features from 58 to 21 while still keeping over 95% of the information. And then we use shared nearest neighbor algorithm to measure the similarities between these neurons and then apply community detection algorithms to identify cell clusters. This process will be repeated thousands of times by random resampling until convergence. So here is our co-clustering matrix for this data set. As you can see, there are 3 major blocks in this data set, one of them corresponding to the thalamic neuron and 2 corresponding to striatum neurons. And within the thalamic block, we see subblocks corresponding to neurons from the VPM and LGN-cores. And for the striatum neurons, these 2 blocks corresponding to projections to substantia nigra and the globus pallidus external. As you can see in these examples, the morphologically distinct cell clusters correspond to different projection patterns, which is one of the key questions we want to answer to begin with. And we are seeking for answers to this question at greater details by quantitative analysis of the distribution of each single neuron across the different brain regions and layers. To this end, accurate data registration is very important, and our team member Yang will introduce his methodologies.
Good morning, everyone. My name is Yang Li from the informatics team. So in this section, I'm going to talk about how we map the neurons from the different mouse into the Allen mouse common coordinate framework. So our first question is, why do we want this mapping? So this is because after the reconstruction, we have a very beautiful data. But this data still live in different spaces, and they cannot talk to each other. This is just because each brain has different size, orientation, and anatomical variations. So by registering them together, we are going to have a way to integrate all this information together. So to the center of the CCF is such a population average template. We computed from over 1,600 mouse brains. So as you can see, it is really rich in anatomical details, and especially it actually shows many of the structures not quite visible in a single brain, for example, the barrel field. And on top of the template, we also have over 600 anatomical structures annotated. That means once the mapping is done, a lot of quantitative analysis will become possible. So this is one example of the FMOST image we have for registration. So the first thing we did in the pre-processing step is, in the frequency domain, we first removed these inhomogeneity strips, and that gave us a much cleaner image. But as you can see, still there are some other kind of noise here and here. So by using the information from the right channel, we keep on making a cleaner image like this. So when the processing is done, what we did is we just stacking this 2D image together. That gave us such a 3D volume. This is actually the input to our registration algorithm. So the registration algorithm actually takes two steps. The first is called the global. That means in this step, we remove the whole brain together. So it just roughly aligns the subject to the template in terms of the size and orientation. And in the following step, it's called a local alignment. That means in this step, we can warp each part of the brain individually. So after this step, as you can see, we have a much better alignment between the subject and the template. And other than this image, actually we have another product from this registration, is this beautiful painting. This is actually the deformation field. So by using this deformation field, we can now map each of the neurons into the CCF space. So as you can see, neurons from different mouse at different locations now live in the same space, especially now in this space, we have already know the boundary of many structures we have interest. That means we can do quantifications. Yeah, so for example, this giant neurons, so we can now ask some questions like, what are the structures it projected to, and how strong this projection is in each of the structures. And also, there's another great tool now available in the CCF space is a cortical coordinate system. So by using the streamlines perpendicular to the cortical layers, we can now project the neurons we just seen in the previous slides first to the top surface of the brain. And then we can do one further step is to unfold the surface. That would give us such a completely 2D planar view. That view allow us to see the whole giant neuron. So for next, Zhi will talk more about the reconstruction and our Allen SEU Center.
Hello everyone, my name is Zhi Zhou from Computational Neural Anatomy Group. So actually one of the goals in this project is trying to reconstruct 60,000 to 600,000 neurons from 600 whole mouse brain over the next 5 years. How can we really achieve this goal? So in my talk, I'm going to introduce our tools and technology, our new facility. At the end, I will also give some update about our achievements so far. So we have in-house very powerful software called VAR3D for 3D image visualization and analysis. But if we really want to handle very large-scale image, like the whole mouse brain data, typically the image size will be between 10 terabyte to 30 terabyte. We develop another big data engine called Terafly. So when we use the Terafly module, we can really efficiently visualize and analyze very large-scale image. So after we get the tool, the next step is how can we really increase the throughput? So we designed the Archer Tracer framework. So in this framework, we can use any existing tracing algorithm to fully automatically reconstruct the single neuron reconstruction at the whole mouse brain level. We also include the AI engine to learn from human being. We can further increase the accuracy of the automatic reconstruction. After we get the reconstruction, we need the human being help to check the result, also give some feedback to correct it. We develop another technology using virtual reality. The annotator can really look at the neuron structure and the reconstruction in the virtual space and then to do the analysis or the correction. Of course, we also provide the data protocol which has been described by Peng before to make sure all the reconstruction we generate are meaningful. So at this stage, we are still trying to develop more algorithm tools and to speed up the entire process. So in here, I just want to show two examples about our technology. So the first one is our automatic reconstruction. In these two examples, we can see we have generated more than 100 automatic reconstruction for each brain. Use our Archer Tracer framework. So if the neuron is sparse labeled, we can generate the automatic reconstruction like this. You can see in purple. Compared with the manual reconstruction, which in light blue, we can see most of the branches has been captured while we are doing the auto tracing. So the second technology I want to mention is the VR technology. Also has been described by Yun before. When the annotator wears the VR equipment, it can immediately visualize the entire whole mouse brain at the global scale. And you can all the way zoom in to the highest resolution to see the single neuron level. You can rotate the image and you can do the annotation. So in the virtual space, actually, the image will be more clear and you can do the annotation more precise. So in the early of this year, we established a new facility called the SEU Allen Joint Center for Neuromorphology, which is in Southeast University in China at Nanjing. So here I'm showing the layout of our center. From the layout, you can really see our annotator and the developer, they're sitting together. They communicate and exchange idea and to improve the entire process of the annotation. We also have the supercomputing facility to provide the service, including network storage. We have big data visualization area, and people can see the whole mouse brain structure, also the reconstruction we generated. So here is two examples. So the first one you can see, for annotators, they are really using our VR equipment to annotate the neuron structure in the virtual space. So especially the two ladies on the right side, they are using our latest technology called the cooperative mode. So in this mode, they are tracing the same neuron image at the same time. After annotators done with the annotation, all the morphology will be immediately showed in our large screen, also with the analysis result. So in this way, you can really see the morphology, see what's the current status of our annotation and what's the quality of the reconstruction we generated. So our center also provides a number of high-end hardware to support the entire annotation process, including this petabyte storage, fast SSD storage, the high-definition tiled display. Also, we have the touch screen. For each annotator, we have very powerful computer and the VR equipment. Total, we have 11 of this right now. So here's some number about our achievements so far. So we have already generated more than 200 full morphology of single mouse neuron at the whole mouse brain level. We're still continue to develop our algorithm tools and technology to really speed up the annotation process in each step. So except the Allen Institute Center, we also have two different groups. One is at Wenzhou in China, another one is at Seattle in Allen Institute. Actually, people from three groups, they are working very hard together trying to produce neuron reconstruction as complete, also as many as possible. So in the end, we also like thank our founder, Mr. Paul Allen, for his support, vision, and encouragement. Thank you very much.
Great. Thank you to the morphology team. We have a few minutes for questions before the break.
This is really amazing, guys. Thanks for doing all this work. Very quick question, maybe to the analysis team. Do you guys start getting a sense for how much variation in the data set in morphological features are explained by individual variation, and are there certain features of morphology that are more robust across individuals versus ones that may be more variable? And do you start sort of getting a bit of a sense for that?
So thank you for your question. We are still at the stage of collecting more and more data, and currently we have been looking at 2 mouse brains, and they have the same genetic modification. So they basically label the same sets of cell types, and we have compared them. Actually, the cell type is more dominant than individual difference, at least according to this data set.
Just to follow up on that question, how variable is it between male versus female mice?
We don't have that data yet, but yeah, we'll look into that in the future probably.
So looking at some of those reconstructions, it looks like the axon typically branches a lot near the soma and then also at the distal end. And I'm assuming that more branching means more connections. And I was just wondering how you quantify where the axon connects.
You mean the connection of the axons?
Yeah, like what regions of the brain it connects with.
The regions we have mapped to the CCF, so we know the brain regions. But if you mean the direct pre or post synaptic connections, from this data we basically don't have that, because we specifically label certain type of cells. It's hard to control whether the next stop is also labeled or not.
Also, can I say one more? I mean, so he also mentioned the level 3 reconstruction, where we can potentially pick up the axonal boutons, and then that would allow us to give some kind of a measure of what the connection strength would be. Yeah.
We have one more question. So regarding the classification again, it seems like when you're using these whole brain, super large scale reconstructions to classify, there's depending how you're choosing your features, there might be some sense in which just simply based on where the region the cell is coming from, the large scale features are going to be different, and you're going to be sort of enforcing that different regions have different cell types, if that makes sense. Have you thought about this question, and could you address that briefly?
Yeah, so the features we collect were coordinate free. So we have, so basically, for example, we measure the length of the dendritic axon, total length and stuff. These are coordinate free. And also before analysis, we have performed also the rotation of the images to ensure that the largest span is by the x-axis. So by doing this rotation and the shifting, the coordinate information is already gone. So it's not like a loop prediction.